Hi there, welcome to this video. So we're going to go through the control variables criteria. So this is M3, which is to explain how control variables have been managed when undertaking the investigation. Now it's really important that this is an explain command word. So you can't just list the control variables, you have to explain how you've tried to keep them controlled. All right. So this is per experiment. So add a paragraph on control variables underneath each method. So maybe under each rationale, add a new heading, control variables, and write a paragraph for each experiment that you did. If you're not sure, even after this video, AI can help with ideas. Do not copy and paste from AI, but it just can give you ideas of things that were controlled in an experiment. So you could copy and paste your method in and ask it to suggest what the control variables might be. So things it could include is things like people. So for example, when you're looking at a reagent test strip, you're looking at a color change, or even the Benedicts and the Biret, you're looking at color change. So it's quite subjective depending on who's looking at it. So making sure it's always you that's making that judgment on the color helps to keep it consistent. So always using the same person. Time, we'll go through them each in turn in a minute, but be specific about times and explain how it's controlled. So the fact that you're using a stopwatch. Temperature, what temperature are we actually looking for, and also how do we control it? So we're using water baths. And volumes, we need to be specific about what volumes we're using and explaining how we're controlling our volumes. So I'm going to go through each experiment in turn with you just to make sure you understand what you could do for each one. So the first experiment that we did was the reagent test strips. So if you think about the method, what we did is we got our little stick and we dipped it into our sample. Yeah, then we pulled it out and we were looking at the colours of those little squares. Okay, so the first thing could be the time that you dipped it. The second thing could be how deep did you dip it in? Deep. Can we write deepness? And then the time before we read or reading the colours. And then comparing to the colour chart. So we'd need to write a little bit about them. You don't have to do all of them for each one. So how long did you dip it in for? Was that in your method? So how many seconds? You need to say how many seconds you dipped it for. Okay. How did you maintain this? Did you just count? Did you use a stopwatch? How did you ensure that this was maintained consistent, controlled each time? Okay. How deep did you dip it? Hopefully you dipped it so all the test squares were submerged. Otherwise, they didn't take in the liquid, so they won't have tested it. So in terms of depth, you need to say that every time you looked carefully and you made sure that it was dipped right under the liquid. And sometimes that meant tipping the pot to the side to make sure each of the squares were underneath the liquid. The time before reading the colours should always be 60 seconds. That's what it said on the bottle that we use. So you need to explain that the control variable was that you always leave it for 50 seconds, 60 seconds before reading the colours off. And you could have taken a photo at 60 seconds to ensure you'd captured what the colour was after exactly 60 seconds. And definitely mention the stopwatch. Then comparing it to the color chart, you need to make sure that you're always keeping the person the same, because different people interpret colors slightly differently. Looking at colors is subjective. So one person might say, oh yeah, I think it's that one. Another person might say, I think it's that one. So it is important that the same person is looking at the color each time. The next test that we did was the qualitative Benedicts. So for this one, what we did is we had a pot of Benedicts, which was a blue liquid. And then what we did is we added that to with a sample of the urine. So we put a certain volume of urine in, we put a certain volume of Benedicts in, and we mixed it, and then we looked at the colour. Okay, so things that we needed to control in that experiment is we needed to control the volume of Benedict's and urine. I think I'm right in saying that we used 2 centimetre cubed of all those. You might want to double check on your method. And the way that we kept that controlled is that we used a Pasteur pipette to measure it. Okay, to make sure that we were always measuring exactly 2 centimetre cubed of the liquids. We then mixed it, so to ensure that we were doing consistent mixing, you could say something like, I inverted the tube 3 times to make sure they were mixed the same each time. And then in terms of looking at the colour, oh, sorry, I forgot you have to heat it. So in here, we heat and then we look at the colour. So in terms of heating it, we put it in a water bath. So to heat it, we should have used a constant temperature. And the way that we controlled the temperature is that we used a water bath that we made by boiling the kettle. And you could say that we always used freshly boiled water to ensure that it was always as close to 100 degrees C as possible. Okay, and then when we looked at the colour at the end, that's slightly subjective, so we need to make sure that we're always using the same person. So it was always you that looked at the colour, and remember, if it's blue, it means there's no glucose. If it was green, there was a low amount. Yellow, there's slightly more. Orange, even more. And if it goes red, or we call it brick red, it means that there's a high concentration. Okay, so just discuss some of those control variables and make sure you say how they're controlled. The next one is very, very similar. The next one that we did, I'm going to just write over this because it's so similar. The next one that we did was called the Biuret test, where we were looking for protein. So again, we used the bottle of solution and we added that solution, which wasn't called Benedict's this time. The solution was called Biuret. So you added your Biuret liquid solution reagent and you added your urine sample. Okay, then you mix them together. You didn't have to heat this one, so the heat bit isn't relevant. You just mix, wait, and then look for the colour. Oh, you could have said on the Benedict's one how long you left it in the water bath and that you timed that for 5 minutes with the stopwatch. So for the control variables then, it's going to be very similar. So how much volume of Biuret did you use? Okay, and how much volume of urine did you use? Again, I think you used 2 centimetre cube, but double check in your method. And you controlled that by using the Pasteur pipette to measure it every time. You needed to make sure you had a consistent mix again. So you could talk about inverting the tube 3 times. We didn't heat this one, so let's take that out. However, you did have to wait. So the time before reading the colour, you could say that you, I don't know if it said a time on your method, but say 5 minutes, and that you measured this using a stopwatch to make sure that it was always controlled. And then when interpreting the colour, again, you need to use the same person looking at the colour because it's slightly subjective if different people look at the colour. But what we were looking for here is if it goes purple, it means that it contains protein. So was it purple or wasn't it protein? Purple. And if we always use the same person, then that's consistent. Okay. The next experiment that we did was all the microbiology. So the first thing you did is to make your streak plate. So the streak plate experiment was the one where you had your microbial cell culture and you used an inoculation loop to transfer a little bit of that into the middle of your agar plate. Oh, sorry, didn't put it in the middle. Let me take that off. You used your inoculation loop and you did a streak plate. So you did like some streaks there, some streaks there, streaks there, and streaks there. Okay, so what things were you trying to do the same every time? So you wanted to make sure that you had the same volume of microbial cell culture. Oh, we didn't measure the volume. But we just used one inoculation loop full. So we should have always got the same amount roughly. We should have done the same streak plate pattern on each one. So they were spread out in the same way, and we just did that by doing that same pattern. You'll be able to get an image of that if you type streak plate on Google. Make sure you reference it if you get a picture. And then we left them to incubate. So we could say the incubation temperature should have been the same. Now we incubated ours at room temperature, which is 21 degrees C. So we left both plates together, so they should have had the same conditions. And then the incubation time should have been kept consistent. So I don't know if you know how long it was. If not, just say 1 week. Yeah, I think that'll be fine for that one. So then you were looking at the selective media, and you filled in your selective media table if you did it on both plates. So you could just say for that, if you're comparing the growth on the two plates, you could say basically it's the same as above. So it's just basically in yeah, I think I would just say for that, just put as above, because you need to have grown them in the same way in able to compare them. So it's a fair comparison. Then you did the colony morphology. So you're looking at the colonies. Oops. So it's important for this one that you looked at the colonies from the same angle each time. The same distance from the plate, like where was your eye compared to the plate? You could say same person doing it, because it is quite subjective. It shouldn't be, but it is because you've not done it many times, so it's quite tricky. You were using the same criteria to look up the features. So we were using that specific language, weren't we? Like punctiform, circular, etc. Okay. So you just need to say this is how you looked at it. Also, you might have, if you used a phone to zoom in or a magnifying glass, you should have used it on both of the plates to make it fair. So always use my phone to zoom in, or I always used a magnifying glass so it was controlled. I always looked at the, the form from above and the elevation from the side. I always made sure my eye was about 10 centimeters from the plate when I was looking. I did the examination of each plate, so it was done by the same person. And I always used the lookup table to look at how to describe the form, elevation, etc. Okay, so things you kept the same. The final experiment that we did was the quantitative Benedict's. Now, this was the most complicated one. We had lots of steps, didn't we? So first of all, we made up our known concentrations. So we had loads of test tubes in the rack, and we had concentrations of 0, 10, 20, 30, 40, and 50. And into those, we added our known glucose concentrations, and we added the quantitative Benedict solution. Which is different to the qualitative one. You get different colour change. Oops, sorry, I spelled that wrong. Oh, it wants to rub the whole lot out. Okay, so what you would then do, just cross that out, is you need to be keeping lots of things controlled as you're adding those. So one thing that you needed to control is the volumes of those liquids. So look in your instructions, in your method, what was the volume? You need to put the actual number in. So you use this many centimetre cubed of quantitative Benedicts, and you use this many centimetre cubed of the known glucose concentrations. So then how did you make sure, put that down, then how did you make sure that you controlled those volumes there? So we used pipettes, didn't we? For the Benedict, we used a graduated pipette. And for the known glucose concentrations, we used something called a Pasteur pipette. That's the squeezy one. So that's how we controlled the volume of the liquids that we were adding. Okay. The next thing we did is we heated these. So we need to talk about the heating process. So you could talk about the temperature. It will have been about 90, 80 degrees C, something like that, because it came out of the kettle, but it won't have still been boiling by the time it was poured out. So how did we try to maintain that temperature? Is what we did is we used a freshly boiled kettle. So we kept reboiling the kettles to make sure the water was as close to 100 as we could get it. Okay, so we made our own little water bath by boiling the kettle and pouring it into a beaker. We tried to keep it hot by freshly boiling the kettle to make sure it was as hot as possible. The next thing we did is we poured these into the little cuvettes, and we put those little square cuvettes into the colorimeter machine. If you remember, there was a little hole and you put your cuvette in there, and then it gave you a reading on the screen. Now there was a few settings on the colorimeter that we needed to control. So we always put the colorimeter on red filter. Okay, and the way we controlled this is that we pressed the red, green, blue button until it displayed red on the screen. So that controlled that. We were looking to record the transmission. So we needed to make sure we were always recording transmission and not recording absorbance. And to control that, we pressed the transmission absorbance button. Okay, until it displayed the T on the screen, and that controlled that. We needed to try and add the same volume of each sample in the cuvettes. So again, we measured that out. We used the Pasteur pipette to transfer it across. Oops. I can't remember how much you added, but you need to add the actual volume, centimetre cubed. Okay, and then you read off the transmission and you filled in your table. So there's lots of things there that we were controlling, and you need to make sure you always say how you were actually controlling those things. Okay, so we should have, in summary, one paragraph on the control variables under each method. You do not need to include all of those things that I've spoken about. As long as you have at least one thing that you controlled per experiment, maybe a couple would be good. All right, that's the end of that video.